2, 3. Hi, my name is Nellie Lin.
And my name is Kershang Chen.
And we are seniors at the Bronx High School of Science, interning at SUNY Downstate Medical Center, Department of Radiation Oncology, and we will be discussing our research project, which involves characterizing a potentially new radioresistance pathway in the endometrial cancer stem cell niche that considers the interplay of HIF1 alpha, COX2, and Wnt signaling cascades.
By identifying this mechanism from which cancer develops radioresistance, we hope to eliminate cancer's resistant properties to radiation therapy, a common treatment undergone by many cancer patients. Our research serves to improve the efficacy of radiation treatment in order to help those with this life-threatening resilient disease.
First and foremost, it is imperative to note that radiation treatment is ineffective because radiation does not target cancer stem cells, which is the source of cancer formation and tumor maintenance, thus allowing the tumor to persist. These cancer stem cells are also inherently radioresistant, serving as a natural radio barrier against radiation.
Once patients are treated with radiation therapy, the tumor initially decreases in size, but after a while the tumor starts proliferating rapidly and becomes further radioresistant.
Our research project serves to understand the core circuitry behind radioresistance in the endometrial cancer stem cell niche and how this tumor mass starts proliferating by studying the Wnt COX2 and HIF1 alpha pathways.
The HIF1 alpha pathway serves to create a hypoxic environment around the cancer stem cell, which means an environment lacking oxygen, and without the presence of oxygen, the effect of radiation therapy may not take place. The COX2 pathway serves to arrest the cell in its cell cycle. In contrast, the Wnt pathway has an opposite effect, which enters the cell into its cell cycle. These three pathways are vital to the core circuitry behind radioresistance.
In our results, we have identified what occurs pre-irradiation and post-irradiation of the expression of Wnt COX2 and HIF1 alpha in the endometrial cancer stem cell niche. This is a pre-irradiation stain for HIF1 alpha as indicated in the green. This shows that there are basal levels of HIF1 alpha present, meaning that the cancer stem cell niche resides in a somewhat hypoxic environment. Pre-irradiation stain for COX2 represented with orange. As you can see from the stain, there is little to no COX2 present, indicated by the lack of orange. This means that cells are not arrested in its cell cycle pre-irradiation. Post-irradiation stain for COX2. COX2 is highly expressed as indicated in the orange. HIF1 alpha is also highly expressed post-irradiation in conjunction with COX2. This is a post-irradiation stain for COX2 and HIF1 alpha merged images. HIF1 alpha and COX2 are both highly expressed, indicating an increased hypoxic environment and a cellular arrest that protects the cancer stem cell from radiation, making it radioresistant. The HIF1 alpha and COX2 are indicated in orange and yellow respectively. They overlap together on the stains of OCT4, which stains for mesenchymal cancer stem cells. This means that mesenchymal cancer stem cells are responsible for the production of HIF1 alpha and COX2. Wnt is highly expressed pre-irradiation and post-irradiation, so it is present regardless of the presence of radiation. As seen in this stain, Wnt overlaps with the NANOX stains, which indicated the epithelial cancer stem cells for which NANOX represents is responsible for Wnt production. The frizzle represents the Wnt targeted frizzle receptor, and the frizzle stains overlap with the OCT4 stains as well as NANOX stains, indicating that the frizzle receptors are on both epithelial and mesenchymal cancer stem cells. We know where the Wnt is being produced, which is the epithelial cancer stem cells, and Wnt is responsible for entering the cell in its cell cycle, making these cells vulnerable to radiation, so epithelial cancer stem cells are radiosensitive. On the other hand, mesenchymal cancer stem cells are the production of HIF1 alpha and COX2, which contribute to cancer stem cells' radioresistant nature, making mesenchymal cancer stem cells radioresistant. Our research project has identified the core circuitry behind radioresistance. When radiation occurs, the cancer stem cells residing in the tumor increases its HIF1 alpha and COX2 production, which protects the cancer stem cell from the effects of radiation. The cells are not vulnerable to the effects of radiation because of these two pathways that make a hypoxic radio barrier and arrest the cell in its cell cycle. Cellular arrest overrides cellular entry into the cell cycle, so Wnt cannot be expressed. When HIF1 alpha completes metabolizing the oxygen, it downregulates, leading to a downregulation of COX-2 through a feedback loop. Therefore, Wnt is allowed to be expressed, entering the cell into its cell cycle, which is responsible for tumor maintenance and tumor progression.
So this entire pathway involving Wnt, COX-2, and HIF-1 alpha was deduced to be the cause of radioresistance and radiation therapies failures. Our discovery of this core circuitry for radioresistance has tremendous clinical significance for enhancing radiotherapy. Using this information, we can make radioresistant cancer stem cells radiosensitive. Go.
Each year, thousands of patients fail treatments because of the inherent radioresistant nature of cancer stem cells. If this pathway can be targeted and the resistant mesenchymal cancer stem cells can be made radiosensitive, countless lives can be saved and treated effectively using radiation therapy.